Episodic memories are stored and restored in different parts of the brain, as neuroscientists have believed since 1970, where the cerebral cortex and hippocampus operate in accordance with a specific system for retrieving memories stored in the brain of events he had previously experienced, as if he were re-experiencing the same event, and if the hippocampus was injured, a person has lost memories for decades, and it was difficult to test this due to insufficient technology at the time. Today, scientists use a new technology to study and treat neurons using light called genetic optics, which is the standard method for exploring brain functions, and depends on the use of mice with genetically modified neurons that shine green when activated, allowing tracking memory formation in the mouse brain, and identification. Thus it has neurons in both the cerebral cortex and the hippocampus that are active during learning and memory recovery. Where the hippocampus recovers memory by reactivating cortical cell patterns that were active during learning to do their job, and to prove this the hippocampus was first disabled, and secondly, marking the neurons that were active during fear with long-term fluorinated protein H2B, GFP, as these proteins allowed the identification of tagged neurons after several days of learning and inhibition by laser stimulation. The memory recovery process is thus interrupted and neither the cerebral cortex nor the amygdala are activated. Thus, inhibition of the hippocampus did not change the size of the total stimulation in most areas of the brain, but it selectively prevented neurons that were active during learning from reactivating them during memory recovery. We conclude from this that the hippocampus reactivates cells expressing specific memories during the process of recalling the memory. After knowing this, how did the scientists wipe the memories from the rat's brain? Initially, the scientists needed to provide the mice with some bad memories, by subjecting them to electric shocks in a cage, which in turn linked this site with frightening memories, and remained static without moving when placed in the cage itself again. The tagged cells allowed the researchers to identify specific cells in the cerebral cortex and the hippocampus that participated in learning and recall frightening experiences, and when these cells were inhibited by light via the optic nerve fibers destined for the mouse brain, the mice were no longer able to remember their fear associated with that cage, and inhibition did not affect other cells unrelated to the hippocampus. It should also be noted that cells can also be activated by light and in a similar manner. Now that the memory is stored and recovered, this optical memory eraser opens new horizons for treating both PTSD or even memory loss treatment.